Hi biologists, let's start by looking at the learning objectives for this lesson. At the end of this section, and following the biology syllabus, you should be able to define the term mitosis, give a simple explanation of, with the help of diagrams, chromosome behaviour during mitosis. Names of stages or chromosome parts are not required. What does this actually mean? What are we trying to understand? Well, basically, it's very straightforward. You just have to know the definition of mitosis. You have to be able to explain, with the help of diagrams, how chromosomes behave during mitosis. The good news is you don't need to know the names of the stages and you don't need to know the chromosome parts. Let's look at mitosis. Knowing the definition of mitosis is a learning objective for this lesson. Therefore, we have to make it our business to learn it off by heart. Mitosis is nuclear division. The nucleus splits in two. So for example, if we have a cell and here is the nucleus here, we can imagine that the cell will split in two. Now this is a rather simplified version of what's going on, but it will do us for the moment. When that cell divides in two, you will end up with two new cells that are absolutely identical to each other. Well, my two there don't look exactly identical, but you get the idea. So in proper science language, mitosis is nuclear division in which one nucleus divides to form two nuclei, each containing the same number of chromosomes with identical genes. In other words, two new genetically identical cells are produced with the exact same number of chromosomes as each other and as the parent cell. Because they are identical to each other, they can also be called clones. Now, while knowing the definition of mitosis is an important learning objective, so too is being able to explain the behaviour of chromosomes during mitosis. But before we go there, let's have a look at another few details to help our understanding. Now, when nuclear division or mitosis is finished, the cell will then divide to form two new cells. We can see that here. If we start with a cell with 4 chromosomes, then that cell will divide to form two new cells, each of which are genetically identical to each other, meaning that we started with a cell with 4 chromosomes, we're ending with 2 cells with 4 chromosomes that are identical to each other. 1, 2, 2 long ones, 3, 4, and exactly the same case here, 1, 2, 2 long ones, 3 and 4. These two new cells, as we've just said, are identical to each other. They are clones and they're identical to the original cell. We must also realise that the cytoplasm and the cell organelles are equally shared between the two cells as well. You might recall from a previous chapter the meaning of the word organelle. An organelle is a structure within a cell. Examples of organelles would be mitochondria and in a plant cell, chloroplasts. So these mitochondria and chloroplasts are equally shared between the two cells that are formed. Also, we must realise that mitosis only occurs in somatic cells. Now that's a new term. Somatic cells is just a science term for normal body cells. Now looking at mitosis again, we've learned in a previous video that mitosis follows the interphase stage of the cell cycle. You might recall from a previous video that the cell cycle is really the story of a cell's life. Let's pop back and have a brief revision of that before we move on. The interphase stage is not part of mitosis. You might recall that there are two stages in the cell cycle or in the story of a cell's life. The first stage is a non-dividing stage, which is called interphase. The cell, remember, spends most of its life in this stage. The second stage is a dividing stage called mitosis. So, as we've just said, mitosis follows the interphase stage of the cell cycle. Again, it is nuclear division. It is division of the nucleus, resulting in the production of two identical daughter cells or daughter nuclei. Now we must realize that mitosis is a continuous process. It's rather like a film. It starts, continues, and then ends. It's not like a show where it is divided up into different parts with a break between each one. For the sake of simplicity and so that we can understand what's going on, it is described as if it had 4 distinct stages. But as we've just said, each stage runs smoothly into the next. Now that we have covered the basics of mitosis and have an understanding of what it's all about, we can now proceed to look at each of the 4 stages and focus on how the chromosomes are behaving, which is actually one of the objectives of this lesson. Stage 1. Well, in stage 1, you can see the chromosomes. They are visible. You can also notice that the nuclear membrane is starting to break down. Again, as we've already said, there are 4 chromosomes in this cell, chromosome 1 and chromosome 2. They were the 2 long ones from earlier on. But this time you might notice that they are double-stranded. It's as if they have photocopied themselves. Again, we have chromosome 3 and we have chromosome 4. And again, they are double-stranded. So in a nutshell, in stage 1, the chromosomes shorten and thicken. They become visible. 2, the chromosomes are double-stranded. 3, the nuclear membrane starts to break down. Make sure that you know the steps involved in each of these stages and are able to draw the accompanying diagram. In stage 2, what differences do we notice? Well, we notice really that the nuclear membrane has broken down, that the chromosomes 1, 2, 3, 4 are starting to line up one after the other. Now, a scientific word for a straight line is an equator. So they line up along the equator is a phrase which is often used here. You also will notice the presence of these spindle fibers that are attached to each of the chromosomes. Coming from the right-hand side, they're actually produced by that structure called a centriole that we don't really need to know that much detail about, and coming from the left-hand side here as well. I like to think of these as ropes. That will help us to understand what goes on in the next phase. So here, the chromosomes move and they line up along or across the middle of the cell. Now, I like to line my chromosomes that way, but there's nothing stopping people lining them up horizontally along the equator of the cell. It's the exact same thing, it's just a matter of drawing the diagram from a different orientation. We can see that here, if we twist the diagram, that we have drawn, and now the chromosomes are going to line up across, but it's the exact same diagram. Fibers will then attach to each of the chromosomes, and the nuclear membrane is fully broken down. And again, as a reminder, remember to learn what happens in each stage and practice the accompanying diagram. Now, what happens in stage 3? How does this story unfold? Well, in stage 3, you might notice that the chromosomes are pulled apart. That's why I like to imagine that the spindle fibers are like ropes and that they yank the two chromosomes, pulling them apart from each other. That's a little bit of a simple picture, but it does help us understand what's going on. Now, those of you who are sharp-eyed might notice that we've 1, 2, 3, 4 chromosomes being pulled to the left, and 1, 2, 3, and 4 chromosomes being pulled to the right. So as a spoiler alert, remember we are hoping to end with 2 identical cells at the end of this process. So in stage 3, the fibers contract and they pull on the chromosomes, they shorten, and they cause the chromosomes to be pulled apart to opposite sides of the cell. Pointing out to you that these chromosomes are identical, so in a nutshell, identical chromosomes or identical genes are going to be pulled to each side of the cell. The last stage in mitosis is stage 4. How does the process unfold in stage 4? Well, in stage 4, our 4 chromosomes will have been pulled to the left, and our 4 chromosomes would have been pulled to the right. So a nuclear membrane will start to reform around the chromosomes on the left, and a nuclear membrane will reform around the chromosomes on the right. You should be able to see the idea now that we have produced 2 identical nuclei with the same number of chromosomes in each one. As mitosis draws to a close, the cytoplasm will start to pinch in between both cells, resulting in the production of 2 new identical cells. So as a reminder, in stage 4, a nuclear membrane reforms around each group of chromosomes. The chromosomes eventually will elongate to become chromatin. In other words, they will get longer and they will become invisible. Do you remember from a previous video? IN in chromatin and IN in invisible. Chromatin is just a phrase used to refer to chromosomes when they are invisible. The cytoplasm will then divide in 2, forming 2 new cells. Again, just to remind you to learn what happens in each phase of mitosis and be able to draw the accompanying diagrams. Practice in your jotter. Finally, what are the results of mitosis? Well, as we keep saying, 2 new daughter cells are formed. These 2 new cells are genetically identical to each other and to the parent cell. Just to mention, in case people are getting confused, the word daughter does not mean female here. It just is a science word meaning 2 new cells. Now, for those of you able for a bit more, haploid cells will produce haploid cells. Diploid cells will produce diploid daughter cells. So, for example, if you start with a nucleus with a single set of chromosomes, none of the chromosomes are matching each other, you will end up with a nucleus that is haploid or contain single chromosomes. If you start with a diploid cell where the chromosomes are in pairs, Recall we had one long one in pink and one long one in blue. So that's a pair of long chromosomes. We have one short chromosome in blue and one short chromosome in pink. So that's another pair of chromosomes. So this cell is actually a diploid cell, starting with pairs of chromosomes. And you will notice here that we end up with two daughter cells where the chromosomes are in pairs, or they are diploid cells. And a final reminder, mitosis only occurs in somatic cells, cells not involved in reproduction, normal body cells. So the stages of mitosis are stage 1, where the nuclear membrane disappears, the chromosomes are visible and they are double-stranded. Stage 2, where the chromosomes start to line up across the center of the cell and spindle fibers are formed. Stage 3, where the chromosomes are pulled apart, pulled to the left and the right hand side of the cell. And stage 4, where a nuclear membrane reforms around these chromosomes, forming two new cells. And there you have it. Practice in a jotter. Do it on the double. Now that we have reached the end of our lesson, have we achieved our objective? Can you define the term mitosis? Can you give a simple explanation of, with the aid of diagrams, chromosome behavior during mitosis?